Tes, tes, tes. Di Zoom-nya nggak masuk. Masuk. Sensitif kok ini orang jadul ini.
Nggak masuk, di YouTube nggak masuk.
Oh. Dok, dok. Masuk ya. Nah. Tes, 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 masih kedengeran? Tes, tes, tes. Benar Pak?
Iya di YouTube sendiri.
Iya Pak, kebanyakan ya.
Langsung dari luar.
Di sini dia nanti ke sini.
Yang boleh.
Boleh, bukan orang Singapura.
Oh, Singapura dia mau kerja
Di sini.
Oh, dia di sini.
Di sini. Di
Oh, oke. Ya.
Kalau tampilan Zoom
Rumah lagi. Bentar. Bentar. Bisa nyuci Pak dari tadi.
Iya, itu aja kan paling itu pasti lamanya. Ya,
Mm-mm. Mm-mm.
Bila
Selamat pagi, Ibu sekalian. Nampaknya suara saya sudah terdengar ya? Ya, sesaat lagi kita akan memulai acara kita. Saya mengucapkan selamat datang bagi para peserta sekalian yang telah bergabung di ruang rapat virtual Zoom maupun yang streaming di channel YouTube kami. Good morning, ladies and gentlemen, distinguished guests and respected professionals. Welcome to the seminar on empowering precision oncology through single-cell technology and anatomical pathology perspective here at the National Cancer Center, Darmais Cancer Hospital. I am David Sitinjak. I will be your host for this event. Selamat pagi Bapak, Ibu, serta sejawat sekalian. Selamat datang di acara seminar Empowering Precision Oncology through Single Cell Technology and Anatomical Perspective yang diadakan di Pusat Kanker Nasional Rumah Sakit Kanker Darmais. Saya Dr. David Sitinjak yang akan memandu acara pada hari ini. Ladies and gentlemen, before we proceed with the first speaker, let us go over a few housekeeping rules to ensure a smooth and productive seminar experience to us. Number one, please ensure your microphone is muted when you are not speaking to minimize the background noises for the online participants. The seminar will be conducted bilingually, meaning presentations and discussion will be presented in Bahasa Indonesia and English. Participants are encouraged to use the language they are most comfortable with and when asking questions or making comments. When asking questions, please state your name and institutions before speaking to help us to have a better understanding of the context and background of the discussion. For the online participants, we kindly request to use raise hands feature to indicate your desire to speak in the chat. And our team will assist you in turning on your microphone. For offline participants, we request that you wait for a microphone to be passed before asking a question or making comments. This will help ensure that everyone can hear and engage in discussion effectively. Bapak Ibu peserta sekalian, saya mengingatkan tata tertib seminar hari ini harap untuk selalu mematikan mikrofon ketika pembicara sedang berbicara dan ketika mengajukan pertanyaan harap mengajukan nama dan institusi sebelum mengajukan pertanyaan. Bagi peserta yang bergabung di ruangan ini, harap menunggu sampai mikrofon disampaikan kepada saudara sebelum menyampaikan komentar atau pertanyaan. Demikian. Now let us proceed to the first speaker. To commence this seminar, please join me in welcoming our esteemed the Director of the National Cancer Networking Program, Darmais Cancer Hospital, Dr. Evelina Susana, to deliver the opening remarks.
Bersama bunga. Situ ya. Selamat pagi. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera, salam sehat buat kita semuanya. Saya mengucapkan terima kasih pada pagi hari ini semua teman sejawat yang ada di seluruh tanah air yang bergerak di bidang patologi anatomi. Kita berkumpul di sini bersama-sama untuk meningkatkan pengetahuan kita tentang bagaimana kecanggihan dalam hal diagnosis. Jadi izinkan saya hari ini mewakili Direktur Utama Rumah Sakit Kanker Darmais beserta jajarannya untuk membuka acara ini, dan beliau menyampaikan salam untuk kita semuanya karena pada waktu yang bersamaan sekarang ini ada penandatanganan dengan 4 gubernur di Kementerian Kesehatan yang semuanya itu berhubungan dengan transformasi layanan kanker di tanah air, termasuk diantaranya adalah transformasi untuk layanan diagnosis dan penetapan stadium, di mana di sini sangat erat hubungannya dengan profesi patologi anatomi. Di sini kami berharap untuk semua teman-teman dengan kesempatan ataupun boleh dikatakan strategi yang akan dilakukan pada saat bertugas itu mengingat kepada penetapan strata dari rumah sakitnya masing-masing. Jadi ada strata paripurna, strata utama maupun strata madya. Namun demikian untuk sistem pengetahuan ataupun peningkatan pengetahuan itu kami akan tetap memberlakukan hal yang sama. Seluruhnya mendapat kesempatan yang sama sehingga pada suatu saat apabila rumah sakit tersebut memerlukan pelayanan yang sudah lebih tinggi tingkatannya, maka akan mudah kita kembangkan bersama-sama. Nah, di sini kami mengucapkan terima kasih juga kepada panitia yang dalam hal ini yang pertama adalah untuk tim pengampu transformasi layanan kanker di Indonesia untuk tim pendidikan, kemudian tim diagnosis dan juga untuk tim layanan kanker yang bekerja sama dengan Diklit Rumah Sakit Kanker Darmais Pusat Kanker Nasional. Sehingga kegiatan ini bisa terselenggara dengan baik. Dan tentu saja kita juga mengucapkan terima kasih kepada pendukung acara ini, yaitu dari 10X Genomic Singapura yang merupakan cabang dari US, dan juga PT Pandu Bioscience yang ada di Indonesia. Jadi dari mereka lah kita akan mendapat pencerahan apa dan bagaimana sistem diagnosis yang terkini, karena kita tahu bahwa perkembangan penyakit kanker sudah sampai di tingkat genetik. Ya, kita sudah tahu sama-sama per definisi bahwa kanker merupakan suatu penyakit di tingkat genetik yang merupakan perubahan dari gen secara kompleks. Sementara kita bekerja di tingkat level itu, sehingga di sini kita berharap kita akan menjadi yang terdepan pada saat pelayanan solid tumor ataupun kanker-kanker padat yang juga ikut serta nantinya bekerja sama secara multidisiplin dengan kolega kita di patologi klinik pada saat nanti menjalankan untuk non-solid tumor. Ya, jadi di sini kita bertanggung jawab penuh untuk yang solid tumor sampai tingkat advance. Ya, jadi dari laboratorium dasar, limited, enhanced, dan maksimum. Dan sekarang ini kita belajar untuk pelayanan kanker yang maksimum. Jadi kami sangat berharap komunikasi kita tetap terbuka, baik untuk pendidikan maupun untuk pelayanan. Jadi kami dengan senang hati di sini akan menginformasikan melalui bidang tim pendidikan dan pelayanan, tim pengampu transformasi layanan kanker di Indonesia. Itu adalah tugas yang kita emban bersama-sama agar layanan kanker di Indonesia dapat berjalan sesuai dengan standar yang telah ditetapkan oleh Badan Kesehatan Dunia, di mana buku Blue Book itu kita pakai sebagai Bible kita pada saat kita melakukan pekerjaan sehari-hari. Mungkin demikian yang kami sampaikan. Untuk itu, we would like to say thank you for the Mr. Shenton, and then I apologize if I make a mistake with the pronunciation. Chongkok Ting, and then also for the team from PT Pandu Bioscience who support all of this activity. Then we will learning from all of you how to implement or how to put this system in the system of the diagnosis in the country. We hope we will work together not in the first this session, but also for the next future. So that's why the diagnosis system for the cancer in Indonesia will follow all of the standard international or worldwide. Once again, thank you very much for your support. Jadi teman-teman sekalian, untuk menyingkat waktu, nanti di sini kita akan belajar bersama-sama dan kami berharap tidak ada keraguan-raguan untuk bertanya, dan kami juga mengerti bahwa kita semuanya ada keterbatasan dalam bahasa sehingga tidak usah khawatir dalam menggunakan bahasa. Jadi kita tidak ada tes TOEFL, semuanya bisa kita komunikasikan dengan baik. Ya, jadi sebatas mereka mengerti, kita mengerti, it means all of the communication deliver very nice. Ya, jadi di situ kita bisa meningkatkan keilmuan kita. Sekali lagi terima kasih untuk kehadiran teman-teman, terima kasih untuk mau meningkatkan sistem layanan kanker di Indonesia, terutama untuk diagnosis, sehingga nanti Indonesia tidak tertinggal dari negara-negara yang lain. Untuk itu saya juga mengucapkan terima kasih kepada Dr. Putri yang menginisiasi acara ini terselenggara dengan baik di Rumah Sakit Kanker Darmais Pusat Kanker Nasional. Akhir kata saya mengucapkan mohon maaf apabila ada kekeliruan atau keterlambatan atau apapun yang mengurangi segala nilai-nilai, tetapi tidak mengurangi maksud kami untuk melakukan kegiatan ini agar terselenggara dengan baik. Terima kasih, selamat mengikuti kegiatan peningkatan ilmu pengetahuan ini, dan kami mengucapkan terima kasih. Wassalamualaikum warahmatullahi wabarakatuh.
Terima kasih atas sambutan yang diberikan, Dr. Evelina Susana. And we proceed to our first speaker, Dr. Dian Cahyanti, Specialist Pathology Anatomic, will enlighten us on strengthening pathology service in Indonesia's health transformation. Please come forward, Dr. Yanti. Dr. Yanti graduated as a medical doctor from Medical Faculty Universitas Diponegoro Semarang and underwent pathology specialist training in Universitas Indonesia. Now has been working as pathologist at Pusat Kanker Nasional, National Cancer Center, Darmais Cancer Hospital. Please.
Oke, terima kasih Dr. David atas perkenalan dan waktu dan kesempatan yang diberikan kepada saya. I will present my session in Bahasa Indonesia, but most of my slides will be presented in English, so I think you can still follow my discussion. Pada kesempatan ini saya diberi amanah, dan tugas untuk memaparkan tentang penguatan layanan patologi anatomik sebagai bagian dari transformasi sistem kesehatan nasional melalui stratifikasi laboratorium patologi berbasis sumber daya. Hal ini terkait dengan tugas kami sebagai salah satu anggota tim dalam pelayanan kanker yang akan menemani sejawat sekalian untuk mencapai standar strata pelayanan kanker sesuai dengan yang ditetapkan. Untuk memenuhi hal ini, selain sarana prasarana, tentunya harus dibarengi dengan kompetensi sumber daya manusia, di mana dalam hal ini akan dipandu oleh Dr. Grace Shalmon untuk pelatihan atau pendidikan dan lain-lainnya yang kita butuhkan untuk memenuhi kompetensi tersebut. Ya, slide. Ya, kanker adalah salah satu penyakit yang menjadi prioritas dalam sistem pelayanan kesehatan nasional, yang merupakan bagian dari salah satu pilar dalam transformasi sistem kesehatan di Indonesia. Nah, kenapa demikian? Karena kanker adalah salah satu dari penyakit katastrofik yang memiliki angka morbiditas dan mortalitas yang masih tinggi. Sehingga diperlukan optimalisasi dari pelayanan rumah sakit dengan cara meningkatkan kemampuan sumber daya manusia, fasilitas, serta dengan memperhatikan tata laksana dari sistem rujukan dalam pelayanan kesehatan yang diberikan. Nah, jadi tanggal 11 Mei lalu telah ditetapkan Kepmenkes tentang rumah sakit di jaring pengampuan pelayanan kanker di Indonesia, yang mana terdiri atas rumah sakit pengampu dan rumah sakit yang diampu. Rumah sakit pengampu adalah rumah sakit dengan strata kemampuan paripurna dan utama. Sedangkan rumah sakit yang diampu adalah rumah sakit dengan strata kemampuan utama dan madya. Nah, SK ini didapat sejawat unduh dari pencarian di internet, jadi dapat memastikan sejawat sekalian bertugas di rumah sakit dengan strata apa. Ya, slide. Ya, jadi yang dimaksud dengan rumah sakit strata madya dalam layanan kanker adalah rumah sakit dengan kemampuan melakukan layanan kanker berupa bedah, terapi sistemik dengan kualifikasi SDM serta fasilitas seperti dalam slide berikut. Ya, seperti dalam seperti dalam slide dan nanti ada di SK tersebut juga. Nah, sedangkan yang dimaksud dengan rumah sakit strata utama dalam layanan kanker adalah rumah sakit dengan kemampuan layanan kanker seperti di strata madya, ditambah dengan layanan radioterapi, disertai dengan kualifikasi SDM dan fasilitas yang sesuai. Sedangkan yang dimaksud dengan rumah sakit paripurna dalam layanan kanker adalah rumah sakit dengan kemampuan melakukan terapi yang komprehensif dan mutakhir. Ya, misalnya microsurgery, kedokteran nuklir, transplant, kartisel, dan proton terapi. Slide berikutnya. Ya, ini adalah pemetaan berdasarkan surat dari Dirjen Yankes tentang regionalisasi pelaksanaan pengampuan. Jadi di nasional akan ada 14 rumah sakit strata paripurna, di mana masing-masing rumah sakit paripurna akan mengampu 2 atau lebih dari rumah sakit strata utama dan beberapa rumah sakit strata madya. Ya, nah ini detailnya ada di SK dari Kemenkes yang baru saja di SK-kan. Ya, slide berikutnya. Ya, slide. Ya, nanti bagaimana seseorang dengan tumor-tumor solid di tubuhnya didiagnosis sebagai kanker adalah merupakan tugas, fungsi, dan kompetensi dari seorang dokter spesialis patologi anatomik yang ditunjang oleh ketersediaan sarana prasarana dan fasilitas untuk itu. Ya, jadi slide berikutnya. Ya, jadi berdasarkan ketersediaan infrastruktur pelayanan kesehatan di suatu rumah sakit, fasilitas diagnostik, dan akses seseorang penderita kanker terhadap terapi, maka layanan laboratorium PA dapat kita bagi menjadi patologi dasar atau basic, limited, enhanced, dan maximum pathology. Ya, kenapa ini dibuat? Ini adalah sebagai utamanya adalah untuk standarisasi ya. Jadi standarisasi jawaban hasil PA. Nah, selain itu dibuat untuk sebagai modalitas kita untuk mengadakan diagnosis sebagai dasar dari terapi kanker dan untuk menetapkan strategi yang tepat dalam layanan kanker. Dan juga sebagai penyedia informasi untuk studi-studi di bidang onkologi. Baik. Ya, jadi yang dimaksud dengan basic patologi adalah sarana yang paling minimal yang harus tersedia untuk menyediakan slide rutin yang akan dianalisis oleh seorang patolog. Ya. Nah, jadi untuk pemeriksaan lanjutan itu dimulai dari limited pathology, di mana suatu laboratorium PA sudah mampu untuk melakukan tes untuk membuat kesimpulan yang definitif. Namun tidak termasuk untuk tes-tes atau uji terhadap marker yang bersifat prognostis atau prediktif, yang di mana rumah sakitnya sendiri belum bisa untuk melakukan terapi tersebut. Nah, jadi untuk yang enhance artinya laboratorium tersebut sudah mampu untuk melakukan pemeriksaan terhadap marker-marker prognosis dan prediktif, dan rumah sakitnya sendiri telah mampu untuk melakukan terapi yang terkait. Namun belum sampai ke pemeriksaan-pemeriksaan yang bersifat molecular assay. Nah, sedangkan untuk yang level maksimum itu sudah mampu melakukan pemeriksaan sampai molecular assay, misalnya PCR, FISH, NGS, dan lain-lain. Ya, slide. Ya. Jadi basic pathology itu adalah layanan patologi anatomik yang sudah harus ada di strata madya sampai kemudian itu udah minimal ya. Jadi madya itu sudah harus sampai limited sebenarnya. Nah, di mana baik spesimen sitologi maupun histologi, luaran dari fase analitiknya adalah slide dengan pewarnaan rutin, hematoksin eosin atau papanicol untuk sitologi yang kemudian akan kita analisis. Ya. Sedangkan yang advanced pathology slide berikutnya merupakan pemeriksaan di level subseluler, ya, di mana sebagai patolog ya kita harus memahami embriogenesis, histogenesis, dan karsinogenesis dari seluruh sistem organ dalam tubuh manusia, sehingga kita juga akan memahami ya dasar dari suatu uji level subseluler yang sudah tersedia, baik berupa manual maupun berupa alat. Jadi kita mampu untuk memahami kasus-kasus yang kita hadapi itu apa. Apakah memiliki indikasi untuk dilanjutkan pemeriksaan IHK? Dan tentunya kita dengan memahami hal tersebut, kita juga akan mampu mengoptimalisasi dan melakukan validasi terhadap suatu protokol, dan memilih panel dan menganalisis dan membuat interpretasi atas panel yang kita buat. Sehingga hasilnya akan didapatkan luaran suatu diagnosis, suatu entitas yang sesuai dengan klasifikasi WHO. Ya, slide 10. Ya. Nah, bagaimana implementasi dari sertifikasi berbasis sumber daya ini? Saat ini ada 3 jenis kanker yang menjadi prioritas di fase pertama ini terkait insiden, morbiditas, dan mortalitasnya yang masih tinggi, yaitu kanker payudara, kanker paru, dan kanker serviks. Nanti ditambah dengan tumor-tumor anak, baik solid dan non-solid, yang mana kita harus bekerja sama juga dengan sejawat patologi klinik dalam rangka menetapkan diagnosis dan strategi pelayanan yang tepat. Ya, jadi ini adalah rekomendasi untuk tumor-tumor payudara. Ya, jadi dalam hal pengampuan layanan kanker, patologi anatomik di Rumah Sakit Madya sudah harus sampai di level limited ya. Jadi artinya sudah mampu melakukan pemeriksaan dan interpretasi dari imunohistokimia. Ya, jadi ini adalah rekomendasi untuk resource certified diagnostik pada tumor-tumor payudara, di mana rumah sakit dengan layanan kanker di level basic sudah mampu secara klinis untuk mendeteksi adanya tumor di payudara melalui pemeriksaan fisik payudara klinis dan melakukan pengambilan sampel baik berupa sitologi aspirasi maupun biopsi core dari tumor-tumor payudara. Dan di sini patologi anatomik sudah kompeten untuk menegakkan diagnosis mulai dari lesi jinak, prekanker, hingga karsinoma invasif melalui sampel yang diperoleh dari tindakan klinis tersebut. Nah, untuk itu sejawat sekalian, di rumah sakit masing-masing sudah harus ada satu pelaporan baik berupa SOP ataupun panduan praktek klinik terhadap komponen apa saja yang harus kita laporkan, baik dari spesimen sitologi, dari biopsi core, dari BCT atau breast conserving treatment, maupun mastektomi, termasuk standar pelaporan pada mastektomi pasca kemoterapi neoadjuvan. Ya. Jadi untuk rumah sakit Strata Madya dalam hal diagnostik sudah harus tersedia sarana untuk pemeriksaan imunohistokimia, untuk konfirmasi subtipe molekuler dari tumor-tumor payudara, untuk kepentingan terapi sistemik, yang mana dengan set manual saja sudah bisa kita kerjakan. Untuk neoplasma paru, algoritma atau bagannya belum bisa ditampilkan seperti tumor-tumor payudara tadi, karena sampai saat ini dasar dari pengambilan keputusan untuk terapi adalah berdasarkan morfologi tumor. Jadi apakah tumor itu merupakan yang small cell atau non small cell. Dan apabila yang kita temukan adalah non small cell, dan untuk kepentingan terapi kita harus bisa membedakan apakah dia squamosa atau kelenjar atau adenokarsinoma ya. Jadi dalam perspektif patologi anatomik, kanker paru memang punya karakter dan permasalahan dan tantangan tersendiri terkait dengan sampel yang kita terima. Sehingga dibutuhkan kerjasama dan peran aktif seorang patolog dalam tim multidisiplin, sehingga kita bisa tetapkan diagnosis yang tepat hingga ke status molekularnya. Slide, ya. Dalam lingkup tumor-tumor paru untuk laboratorium patologi anatomi dengan fasilitas limited hingga enhanced, dibutuhkan ketersediaan antibodi seperti yang dalam slide ini, yaitu P63, P40, dan CK56 untuk karsinoma sel squamosa, dan TTF1, napsin, dan CK7 untuk adenokarsinoma. Namun berdasarkan referensi dan pengalaman empiris di TPA Darmais, untuk spesimen yang terbatas, maka panel yang kita pilih yang sensitivitas sudah cukup baik untuk membedakan apakah dia squamous atau non-squamous adalah P63, TTF1, dan napsin A. Itu adalah panel yang paling minimal yang harus kita lakukan untuk membedakan apakah dia KSS atau adenokarsinoma. Ya. Dalam slide ini adalah contoh kasus adenokarsinoma dari biopsi tumor primer paru ya. Sebenarnya dalam hal ini morfologi pada kasus ini secara morfologi dari sediaan rutin kita sudah bisa menegakkan diagnosis sebagai adenokarsinoma dengan pola papiler ya. Nah, kemudian untuk kasus-kasus small cell, kita membutuhkan panel minimal CD56, kromogranin, sinautopisin, dan Ki67. Ya, ini slide ini adalah contoh dari kasus sitologi dan histopatologi dari small cell carcinoma. Ya, ini yang terpulas adalah CD56. Karena sediaannya sangat terbatas, jadi panel yang kita ambil hanya satu. Namun berdasarkan klinis dan seromarker yang lain sudah dapat menegakkan dan untuk melanjutkan terapi sebagai karsinoma sel kecil. Ya. Nah, untuk laboratorium PA dengan fasilitas di level enhance ke maksimum, pemeriksaan lanjutan pada karsinoma paru yang non small cell untuk kepentingan terapi, yang harus tersedia adalah pemeriksaan mutasi EGFR, kemudian PD-L1, ALK, dan ROS1 apabila obat untuk itu sudah tersedia. Ya, ini adalah bagian rekomendasi dari pemeriksaan lanjutan untuk kasus-kasus yang non small cell. Ya, namun dengan adanya kebijakan BPJS yang baru, maka pemeriksaan mutasi EGFR itu sudah merupakan refleks tes dari masa paru yang terkonfirmasi sebagai non small cell carcinoma. Namun nantinya juga akan tergantung juga dari kebijakan rumah sakit masing-masing, PPK, dan clinical pathway-nya. Ini yang concern yang ketiga, yaitu dari kanker serviks, di mana berdasarkan ketersediaan infrastruktur, jadi pemeriksaan lanjutan dari inisial diagnosis pada spesimen biopsi dan maupun reseksi adalah ulasan terhadap P16 untuk menentukan status HPV, apakah dia nanti associated atau yang non-associated HPV, kemudian juga untuk PD-L1 pada kasus-kasus CA cervix yang persisten, recurrent, atau metastasis ya. Jadi untuk interpretasi PD-L1 ini memang akan berbeda-beda, tergantung juga dari klonnya, dan juga apakah nanti dia di breast atau di paru atau di cervix, itu nanti kita akan berlatih untuk itu. Nah, jadi ya sebagai take home message bahwa resource stratified approach ini kita gunakan berdasarkan ketersediaan infrastruktur dari layanan kesehatan, fasilitas diagnostik, dan akses terapi obat untuk pasien-pasien kanker. Dan sertifikasi ini adalah juga yang penting untuk memberikan saran tentang diagnosis, pengobatan, dan strategi untuk layanan kanker. Dan yang penting adalah untuk seragaman interpretasi hasil. Ya, jadi untuk mencapai keseragaman ini tentu saja diperlukan mutlak diperlukan suatu standar pelaporan. Ya, dan untuk mencapai ini semua, selain dukungan fasilitas, tentu saja kita membutuhkan sumber daya yang kompeten, ya, dan membutuhkan pendidikan berkelanjutan. Demikian dari saya, terima kasih. Waktu dan kesempatan saya kembalikan ke Dr. David.
Terima kasih Dr. Dian Cahyanti atas pemaparan yang sudah disampaikan. Kita akan lanjut ke sesi berikutnya. Sesi tanya jawab dan diskusi akan dilangsungkan setelah ketiga pembicara selesai memaparkan paparannya. Next, we have Dr. Sean Tan, who will provide us with introduction to single-cell technology. Before we welcome Dr. Tan to the screen, I will introduce him first. Dr. Sean Tan received his PhD in biological science from National University of Singapore, investigating mechanosensing molecule influence on biological system. After graduating, he worked as a research associate at Mechanobiology Institute in Singapore before joining BioRad as a field application specialist, where he spent 3 years supporting both proteomics and genomics applications. Thereafter, he moved to bionanogenomics, supporting non-sequencing based genome mapping application in APAC region. In October 2018, Sean joined 10x Genomics as a field application scientist based in Singapore. We welcome you, Dr. Sean Tan, to join us to the screen.
Pointer, yeah, this is.
Oh, thank you. So I can move forward. This is not my slide. It's another one. Another slide, not this one. The one will be presented after my session by my colleague Leo. It's okay, I'll stay here. Okay. Yes, this is correct. Thank you, everyone. Selamat pagi. Terima kasih. Thank you for hosting us, 10X, as well as Pandu Bioscience, for arranging this together with Damais. We are very happy to be here today. I've never been here, so it's quite exciting to visit new places for me. So without further ado, I'd just like to give a quick introduction. My name is Sean, and my colleague Leo will be presenting after my session. So here together with us, we also have Copting from 10X Genomics. So Copting actually works with the sales teams in Pandu Bioscience. If you have any questions with regards to products, technology, feel free to ask us during the Q&A sessions or after the session today. So for my session, I'm just going to give a quick overview of what are the products that we have at 10X and how it can drive science as well as discovery forward. So at 10X, we have gone quite a number of years, and over the couple of like 7, 8, or even 10 years, I've been with 10X for almost 5 years, and we have come with products from chromium single cells to it doesn't work. From chromium single cells, which you do with single cells resolutions, instead of doing bulk sequencing, to look at the samples of interest, like you're looking at a smoothie. Instead of just seeing it as a smoothie, you can actually use single cell technology to identify the different fruits that's been used to make your smoothie. And we also have another product that is what we call the Visium Spatial, that allows you to map transcriptome data into the spatial location of the morphology that you're seeing of each tissue section. Last but not least, which is launched recently, late last year, which is our Xenium in situ product. So this product allows you not just to visualize the transcriptome information inside the tissue and know where they are mapped, it is at a single cell resolution. So first, let me just give a quick introduction to what we have in chromium single cell portfolio. So I just want to highlight that this single cell itself, we do allow the user to discover and analyze the individual cell's single cell transcriptome, which is the RNA profile. And we do have immune profiling, where you can actually find out which are the TCR, PCR chronotype. You can find out the CDR3 region. You can find the entire full-length VDJ sequence, which is essential, critical to identify the antigens that it's reacting with. So every T cells, B cells, they have their own TCR receptors, B cell receptors. And with single cell technology, you can actually determine and sequence it out, find out the entire sequence as well as the entire gene expression profile for that particular cell. So you're actually getting multiple data information. But let me just move on. How do we achieve the single cell resolution with 10X technology? So this is a high-level overview of our single cell workflow. So it started with the samples that's been prepared in a single cell suspension. So it can be, the single cell suspension can be obtained through either circulating tumor cells, CTCs, or it can be your blood cells, it can be isolated PBMC, or it can also be cultured cells if you are interested in doing some 3D organoids, or even with, more importantly, in a hospital, you do not get cells except PBMCs, but what you're getting is mostly tumor samples or biopsy samples that you suspect it could be cancerous, and you want to determine what kind of cancer is it, how malignant is it, how invasive, whether is there any aggressive gene transcript in there. You can actually dissociate them into single cells, or if it's frozen, you can also get the single nuclei to profile the entire transcriptome. So what you can do is actually putting the, I'm so sorry for those who are online, because you will not be able to see my laser pointer. So it started with the cells being prepared, mixed with the enzyme, loaded onto a chip that 10x developed. It's called a chromium chip, and it will have multiple rows of wells, and connecting each well is microfluidics channels. So it's like a reservoir with a lot of drains inside that leads to one point. Each chip itself allows you to run up to 8 samples independently without affecting it. So your samples will never mix in a single channel. So once that is done, how do we achieve the single cell resolution is using 10x bar-coded gel bit. Each gel bit contains millions of oligos, which I'll show you in the next slide. And each oligos itself has specific 10x barcode sequence in that oligo. So with the enzyme and cells that is loaded together with the 10x barcode chip into a separate well, it will be placed into what we call the chromium system. It could be a chromium controller, chromium X system that is from 10x, and you'll apply a pressure to drive the gel bits as well as the cells plus the enzyme into the microfluidic channels to form emulsions. Emulsions are basically aqueous droplets that are separated in a partitioning oil. So from there, we have individual droplets that contain cells and gel bit, and therefore you can undergo reverse transcription to barcode it using the oligos that comes from the 10x barcode. So you have individual PCR reactions happening in one single tube. There will be about 100,000 gems. 10% of it will contain cells, up to 10% will contain cells, where you will reverse transcribe, get the transcriptome profile, and generate a library for sequencing. From sequencing, you get your bioinformatics to analyze the data using 10x software, and they could even break it down further, understand how the cell profile relate to maybe trajectory analysis, or even which are the cancer cells, which are not the cancer cells. When you isolate a biopsy itself, there are some cancer tissue, there are some non-cancer tissue. When you perform bulk tissue analysis using sequencing, everything gets mixed in. So you can't really pull out the transcriptome profile from the cancer cells. But with single cell technology, because these are going to be at single droplet and you have every cell's profile individually tagged, you will be able to identify which cells are cancer cells, which cells are normal cells. And since they are normal cells, you can use that as a control to compare what's the transcriptome profile for the cancer cells to the normal cells, and determine whether how to develop therapeutic drug targets to use with your small molecules therapies, immunotherapies that you're working with to see whether they'll work better to cure off the cancer. Because sometimes some markers, it could be a small pool of cancer cells, but the signal is weak among the pool of a lot of normal cells, so they get drowned out. So you thought that, oh, actually the certain gene, like gene X, is not elevated. But in reality, when you take a look at the single cell resolution, you can see that gene X is actually highly elevated in those small pool of cancer cells that is found in your tissue sample. So how do we achieve the barcoding? Because in every gel bit itself, it contains millions of oligos. Depending on the product that you're using, some of them have one type of oligo, some of them have 3 types as you see here. But more importantly, in each of these oligo itself, coming from a single piece, all the oligos contains a common sequence called 10x barcode, which is 16 nucleotide long. And because the gel bit is only together with one cell, one gel bit, one cell, all the transcript that is reverse transcribed that is made into cDNA and made into a NGS library will be barcoded with the same 10x barcode. So if we take a look at the different droplet, you will have a different 10x barcode sequence over there. Therefore, when you are analyzing using the cell ranger pipeline from 10x, you will be able to separate out the transcript that belongs to cell 1, belongs to cell 2, belongs to cell 3, all the way in a single reaction up to even cell 30,000, 128,000. So that is achievable with 10x technology. So I earlier on mentioned about the microfluidics chips, which is the chromium chip from 10x. So here it shows a chromium chip that is placed in the secondary holder that allows you to run up to 8 samples. Each samples, you can actually target up to 10,000 cells per channel, and it only takes 80 minutes to generate the droplet. But this is for droplet generation. The whole entire workflow will take about a day to 2 days to complete to get your NGS library. So for single cell technology itself, usually we are more concerned about multiplets. Therefore, we're actually limiting to 10,000. But we have other products in the single cell portfolio that allows you to target up to, in a single reaction, 128,000 cells, which I will share subsequently. So what is this droplet emulsion that I've been mentioning earlier on? Well, here is a picture to show you what we are expecting to see when we form emulsion or droplets. The GEM itself actually stands for gel-based in emulsion. It means that this is a gel bed that is functionalized with millions of oligos around it. And surrounding in this medium, this little droplet is actually an aqueous droplet that contains all the reverse transcription enzyme, salt, buffer, primers that's inside. During the reverse transcription process, whereby it converts RNA to cDNA, the gel bed will release all the oligos and the cells will lyse, releasing the RNA content and therefore allowing the reaction to be contained inside one droplet for one cell. So you allow you to do the reverse transcription. It's like separating humans into individual cubicles when we enter the toilet. We don't just use one big cubicle with 100 people squeezing in, because that is about. Here, you're actually looking at one cell. Everyone has his own room. So how does the microfluidics flow? Just to take a look at this little video, do you notice that from this channel, there was a cell that came in? So most of the gem will be empty gems, gel-based emulsion, and sometimes there'll be a cell that went in, and that is where we generate the cell's transcriptome profile. From the 10x solution, we have a lot of product that allows you to do gene expression, TCR chronotype, BCR chronotype, VDJ analysis. You can also do epigenomics, looking at open chromatin accessibility. And that is through ATAC, which is using accessible transpose assays. And we also allow users who are more adventurous, who wants to get the gene expression profile, at the same time also get the ATAC to study open chromatin studies. Okay. Using the Multiome kit itself. And last but not least, the fixed RNA. So what does each product allows you to study? Every product has its own focus. One of them is the single cell 3'. It allows you to measure not just the transcriptome, because we actually like multiple analysis. It's all about multiomic nowadays. You do not just want to get one profile. You are not also studying one gene from one sample. You're trying to do multiplexing so that you can understand the biology, the cancer, any disease that you're studying to be more interested. So from the single cell gene expression product, you will be enabled, empowered to interrogate the cell further to understand not just the gene expression, but also cell surface protein, which can target up to as many as what total-seq, which is our partner, can sell you. I heard of people who do 140 total-seq B antibodies, and they are studying the perturbations, the changes of the cell surface protein on different cell types. And you can also perform this CRISPR analysis. CRISPR analysis is highly critical in developing drug development, because you want to target certain genes to see whether knockdown, knock-in, knockout, or elevate the expression using CRISPR, because it's stable. It can be transient, of course, but to study them, you require a lot of work. With 10x, you can study the perturbations that is caused by the CRISPR's knock-in, knockout effect on a cell, in a 10,000 cells, in a single reaction, in a single analysis. So it shows how powerful it is to move science forward, because science is no longer just about doing more studies. It's also about the throughput. So here you're actually doing a 10,000 cells analysis here. But of course, with the infection, sometimes the MOI, when you're doing the CRISPR, you also need to keep it low in order not to have multiple CRISPR introduced into a single cell. But there are also interest in introducing multiple genes to affect when you're performing CRISPR, and that can be done because you know which guide has been introduced into the cell. For immune profiling, you can do the same as whatever I said earlier on, but more importantly, it allows you to sequence out the VDJ sequence, the full-length VDJ sequence for the T cells, B cells, so you know which other chronotype that has expanded or that are even exhausted in the sample that you have collected. And typically people who run this will be looking at the immune cells in PBMCs. It could be also in cancers, cells where they will actually try to enrich the T cells or run the tissues together with the T cells. And how do you know there's a lot of T cells, B cells? Usually they will do an IHC, they will do an IFC, they will just to check using a protein to identify, and then subsequently they use the same tissue samples, dissociate into single cells, and perform the single cells data analysis or experiment. ATAC-wise, so what I mean by open chromatin here is referring to the DNA that is actually uncoiled from the histone complex. So most of the time, DNA are tightly wound supercoiled with histone complex. So they are not really accessible to any of our enzyme that's produced in our body. Right. So when they open up is when gene expression could happen, whereby they could be transcribed into RNA and made into proteins or even other RNA that has functional things to do in the body. So in ATAC itself, some users, researchers, especially in the academic side, they are really keen on finding out how they can regulate the biology, the disease progressions or treatment using epigenetic studies itself. Understanding how the DNA, how to actually even block, to introduce repressor or even to cut out one of the genes using CRISPRs or immunotherapies. So these are some of the assays that you could be embarking on with the ATAC. And with the multiome gene expression, it also allows you to generate the gene expression profile to see how well it correlates to the open chromatin profile that you have obtained. Last but not least is the fixed RNA profiling. Fixed RNA profiling assays is also known as the FLEX assay from 10x. This is actually a relatively new product from 10x that was launched last year. It allows customer or users to be able to arrest the biology at the time of sample collection, because you fix the tissue using 4% PFA or 4% or 10% formalin. And that helps to stop all the supposedly 99.999%, never 100% in science. It's always never 100%. So 99.999% of the biological activities, the biochemical processes inside the biological system. You can actually fix tissues, frozen tissues, fresh frozen tissues, or even samples that are in suspensions, such as PBMC. So you can actually fix it with the 10x Flex sample preparation kit that will require, again, 4% PFA together with some of our enhancer. And you do not just fix it. After fixation, you can store it for a period of time, a couple of months before you get sufficient samples to proceed on to perform single cell data analysis. And this is a high level overview. For this Flex product, what is so different compared to the earlier transcriptome as single cell assays that I mentioned earlier on? For the earlier single cell assays, what we are capturing is polyadenated full-length mRNA, and we're generating full-length cDNA to make it a library for sequencing and then to analyze the transcriptome profile. But here, because of the nature of fixation, which tend to damage the RNA, we are using a probe-based approach whereby we design up to 20,000 probes, up to 20,000 probes that will target up to 20,000 probes that target up to 20,000 genes in your biological system. And the probe will be hybridized overnight together with the samples. And once it's hybridized, there'll be a ligation step, and then continue with the gem generations and library construction subsequently. So what is unique about this step is there are several stop points starting from the initial part of sample collection where you can fix and store. You can also do a hybridizations and then use part of the sample that has been hybridized with the probe to proceed on with the single cell gem generation, and whatever that is left over, you can store them for further data analysis for other assays, other experiments. That is also possible. And you can also multiplex. But with the singleplex assay, when you're doing the flex profile for the singleplex assay, you can also study cell surface protein perturbations with this solution. For the multiplex, it allows you to multiplex up to 16 samples in a single reaction using the different probe sets. There are 16 probe sets with probe ID that will be able to separate it out the sample. So you are separating previously based on gel bit, now it's gel bit plus probe ID. So this is what I meant by the probe set. 10x developed the probe set using a dual probe system, whereby each probe pair targets a 50 base pair sequence on the RNA, so that you can hybridize, ligate, and then continue to be captured by the gel bit and make a library subsequently. What's more important here is the probe barcode ID as well as the 10x barcode ID. So if you imagine you have 16 sets of probe that is used for 16 samples, each sample will have a unique barcode. Any transcript, any genes that it has been bound for that particular sample set, you will have the same barcode, probe barcode ID. So we know, even though I have 5 cells that is in a single gem, I will be able to separate out the transcriptome profile of those 5 cells separately using the barcode, together with the 10x barcode and the probe barcode. So it's pretty massive work that you can done in a single reaction. A bulk reaction can give you 1 million cells, but then again, you can't really resolve it into a single cell. It's like looking at a 720 resolution image compared to a ultra high definition. You see the difference. It gets better appreciated when I got older, honestly. In the past, I don't mind the resolution, but as I get older, the higher definition really helps a lot. So I mentioned about high throughput. So for the single cell assays, earlier on, I mentioned that most of the assays only allows you to target up to 10 cells per channel. We do have the capability to upscale it using the high throughput assay, which is currently available for the 3' single cells or the immune profiling. So you can actually target up to double the number of cells in a single channel. And if you're working with another product, that's what we call CellPlex. It's different from the Flex assay. It's just tagging using a lipid base to tag the samples. You can actually target up to 60,000. And this is enabled on the Chromium X system, which is also launched recently in in the past recent years. I think it's about 3 years ago. Yeah. And that allows you to have, if you were to run the whole entire chip, you can have as much as 4 times the number of cells in terms of throughput. That's a lot of data to analyze, and that should be able to give a better prognosis and understanding of the biological system. So for sample-wise, what is needed? I'm just going to share with you 3 points. And plus 1 more. So of course, it has to be.
Itu. Ketika saya menjalani blok kardiovaskular, nah itu saya merasa saya excited untuk mendalami ini, dan saat itu memang saya sempat mengikuti lomba jadi IMO yang diadakan Fakultas Kedokteran.
Okay, thank you so much for the IT for muting it. So I was saying, the more of the thing is you need to have single cell suspension. It can be single cell, it can be nuclei. Because if you're working with frozen tissue that can be stored for transport or long-term storage in -80 liquid nitrogen, those samples are in a way compromised. So you need to actually isolate nuclei instead of using the intact cell. It's very challenging to dissociate frozen cell. However, at the end of the day, once you get a good suspension, it has to be clean, free of aggregates, free of floating debris, which is not visible to the eye. So what we recommend is usually washing, as well as passing it through a cell strainer. And the cells has to be viable. So at 10x, we actually recommend minimum, try to get at least 70% of the cells to be viable. And that can be done using viable stains such as Darby, Hosh stain, sorry, not Hosh stain, PI. So it can be AOPI, which is a dual stain that detect live and dead cells, or you can just use Trifon blue. And it has to be intact, of course. It cannot be compromised. Imagine bringing back a bag of mee soto, and then the bag has hole. Or the soup comes out, all you get is just mee, no more soto. I think soto is soup, is it? Yeah, soto is soup, yeah. So only left with mee. And you do not want to analyze just whatever content in there, because back then, probably all the RNAs, protease, everything will be kicked in. So you want mee soto, not just mee. So always ensure that the cells that you're working with is intact. I hope. Not moving. Can you click on the slide? Yeah, click on the slide. Okay, now I can move. Yeah, because the control was moved on to the participant window earlier. So now it should work. Okay. So always understand what is your objective, what kind of assay you are going to use. If you are going to perform a IHC, you need a microscope, you don't need a handphone, right? So you need to know what are the tools that you have in order to analyze so that you can actually provide the good data set to generate the good output. Likewise, what are the inputs that can be added in to use? If you are working with open chromatin accessibility trying to study using ATAC studies, you need to use nuclei, not whole cells. If you want to study TCR, PCR, and cell surface protein, you should be using intact cells, not using nuclei. Because nuclei means the cell membrane has been removed. There will be no cell surface protein for the antibodies to tack on. So next, I'll move forward to the next product, which is spatial transcriptomic that is under the Visium portfolio. Just to give a quick snapshot that we do have assays that is require an instrument or does not require an instrument, but both assays, both platforms allows you to work with fresh frozen tissues, which is snap frozen, or that is embedded in OCT, so for those who are familiar with the cryosectioning, that is what you're using, and for FFPE samples, that is with the microtome, and that is basically fixed paraffin embedded. And it is available on not just the direct placement without the machine, or with the CytAssist platform. So when I talk about spatial transcriptomics, I'd like to also share with you what kind of information can you get. This is probably some part that is going to be a lot more interesting to the audience here, because I believe that you all get a lot of tissue samples, right? Biopsies that has been collected, post-mortem samples maybe, to study how the disease progress. Unfortunately, in those cases, the patient are probably dead because it's a post-mortem sample. But we should think on the positive side, they are still contributing to medical science. So what can you get when you do spatial transcriptome? You get the transcriptome profile. You can get the gene expression profile, but you also get the morphology, the histology, the appearance. You can also perform IFC, immunofluorescent chemistry or immunofluorescent staining, to determine where are the immune cells, where are the cancer cells, what are the cell types that's in there, and combine with the transcriptome profile. Because we will be capturing an image of the histological image, whether it's immunofluorescent or H&E stain or other, no, only H&E and immunofluorescent for this platform, we can actually map it back. We can map it back, the transcriptome profile, to this spot. And how is this done? Because we are placing the tissue on a Visium slide. So on a Visium slide, there are spotted oligo. Every spot itself is 55 micron in diameter. I'm so sorry that I forgot to put in that slide here. But if you need further information on the slide design, do reach out to Rabiu from Handu Bioscience. He will definitely follow up with you with the slides that shows you what are the structures. But what I'm saying about the slides here, it actually contains oligos too. So the oligos will be used to capture the barcoded, to capture the ligated probe. So here is also a probe base. I'm just going to talk about CytAssist, which everything will be using a probe-based approach rather than a poly-A tail capture method. So probe-based approach is very similar to the flex, whereby the probe is designed, is a pair that is designed to target one gene. Just one gene. So there will be about 50,000 probe pairs that's been designed to target 20,000, up to 20,000 genes. And it is only compatible with human or mouse samples. But here, hospital, of course, you all treat human, right? I hope so. Human, okay, so it's human sample. This is definitely something that you can use. So what it does is you section it onto a regular positive charge slide, perform deparaffinization for FFPE sample, followed by staining, do the imaging, decrosslink. If you are performing immunofluorescent staining, it will be the cross-link first, so that the epitopes of the proteins is not exposed for the antibody to attach to, or you can also source for antibody that is not affected by the cross-linking. There are limited number of antibody that does that, but it's not impossible. Followed by the imaging, and then that is when we have the morphology, the histology image, and then we continue on with the overnight hybridization of the probe panel that you'll be using. Once that is completed, it will be used to transfer on using the site assist instrument. So the site assist instrument is this on the right side, this little tiny instrument that is narrower than the monitor that we see in front of us, or in fact, it's narrower than this laptop. So it takes up a very small footprint on the lab bench. And this is continued after it has been captured, it's transferred onto the Visium slide. The Visium slide has spotted oligos. Each spot 55 micron, contains millions of oligos. Again, all the oligos on that spot itself will share the same spot ID. You will share the same spot ID. So any transcriptome that is released from the tissue, it will be captured directly by the oligo that is underneath that region on that spot. And it will be barcoded with the 10x spot ID barcode. And that will be made into a library, and we can analyze it on the data. So regardless whether you're working with FFP samples, fixed frozen samples, or even with fresh frozen samples, the workflow might look different because of the staining method, the cross-linking method at the beginning, as well as the instrument to use for sectioning. The end of it remains the same, from hybridization to transfer, to capture, to library construction. And it's used the same kit to do. You do not need to buy new kit, even if you're changing your sample types. And here is the sample requirement with regards to whether you are working with FFP, fresh frozen, or fixed frozen. There will be some QC that needs to be done. Of course, the intactness of the tissue by doing sectioning and then checking it with H&E stain or even with IF. But if you are looking at the RNA quality, what we recommend for fixed samples, which is FFP or fixed frozen, it will be using the DV200 value of 30% and 50% respectively. For fresh frozen sample, it will be a RIN value of greater than 4. So what are these values actually indicating? Is to tell you how intact the RNA transcriptome is. It just tells you the higher the value, the better the intactness of the transcriptome. So last session is just another 2-3 minutes. It's Xenium Institute. This product is launched at the end of last year, and we have several placements in Asia Pacific currently. It is very exciting because it allows you to get the spatial morphology and also get the transcriptome profile. Although it is a targeted transcriptome profile, because it does take a lot of work. It's like doing multiplex IFC. You have to do a lot of imaging done, and it is completely independent of sequencing. So the machine itself will give you the readout, and you just need to run, and the machine will also perform the analysis to give you the initial results. So you do not need any sequencing to be performed. It is this machine on the left. So it allows users to get single cell resolution of the targeted panel of genes that you're interested. Currently, we have released 2 panels, the human breast panel, as well as the mouse brain panel, and each of them can target up to 400 genes with slightly below 300 pre-designed genes, and you can actually add 100 customized genes indeed. And usually these are genes that are of interest that you really want to analyze multiple samples to see how they actually react to a certain treatment, or whether is there a certain target that is showing up from maybe stage 1 to stage 4 of the cancer, or you can do the gradings too. So the whole workflow starts off with doing a sectioning onto the Xenium slides, and thereafter it will actually perform the paraffinization, permeabilization, probe hybridization, and undergo amplification. Once that is done, everything else will be run on the Xenium instrument, where you will do the readout, the decoding, whereby you will be trying to do a lot of fluorescent imaging to identify which are the probe that has successfully hybridized in each of the cells of the tissue. and followed by data visualization. So the probe set itself is tunable. So for the customer, we can actually adjust the number of probes that targets the certain genes, and it's using the padlock design for the probe. And how it generates the amplified is actually using rolling circles amplification to produce rolling circle products. So this is a better diagram to share with you. How does the padlock probe work? Whereby the recognition sequence to the targeted RNA site is contained on the 3' and the 5' end. So it will form a padlock in this way. It will ligate and then followed by rolling circles amplification to generate the amplicons, and that will be decoded on the Xenium instrument. So here is the current panelist, and we are also expecting more release. Why are we releasing tissue-specific panel? It is because you want to specifically target the genes of interest that is more of interest in a particular disease, because the genes that is regulated in breast cancer, in colon cancer, they are all different. It could be in the nasopharyngeal carcinoma is also different, right? There are different targets. Rather than having to do another 28,000 genes profiling, this can be a massive. It could even take you like 4 weeks just to run the decoding. That may not be so feasible. So how long does this whole entire process take? With the current panel itself, with the gene human breast, human brain, if we were to select the whole entire effective area for analysis for 2 slides, it just take within 1 week, you will be able to get the data out. Because the wet lab workflow, which I mentioned earlier on here, this part, simply takes about 2 days work, excluding sectioning placement. With sectioning placement, 1 day plus 1 day, so it's 3 days, the rest will be done on the machine. So that's all for my session. Thank you for your time. My apology for overrunning this part.
Thank you. Give applause. And we may proceed to the next speaker. I will invite Dr. Leo Chan to take up the screen. Before I will introduce Dr. Chan. Okay, Dr. Leo Chan is Senior Manager, Science and Technology Advisor at 10x Genomics. Leo graduated in 2009 with a PhD degree in Marine Biosciences from the University of Delaware in the States. He conducted his postdoctoral research studies at the University of Georgia, Department of Energy's Joint Genome Institute, JGI. He worked at the JGI and Wapergen Biosystems before joining 10x in March 2017. Please.
Great. So hi, everyone. My name is Leo. I'm actually based in the 10x headquarters in California. We're in Pleasanton, next to San Francisco. So because of the time, I'm just going to give you a brief introduction of specifically how the single cell and spatial products are used for cancer research. Now, so I'm going to start with this slide. So we all know that cancer is extremely complex. So to understand cancer biology, you do need to use the advanced tools, such as those from 10x, the single cell and the spatial applications, to really understand what is happening on the tissues. Now, so I'm not going to talk about this. My colleague Sean had just introduced the 10x tree platforms. We have the Chromium single cell and the Visium spatial technology. These are going to be focused today. So for the single cell assay, in theory, is quite simple and elegant. So what you do is a piece of cancer tissues. My goal is to look at what cell types and biomarkers are present in the tissues. So you take the tissue out, dissociate it, I mean chop it off, and then isolate the single cell or single nuclei. Most of the people, they want to do gene expression study as a way to look at cell type classifications and look at biomarkers. And then on the Visium, so many of you are pathologists. So you would like to look at, for example, staining of the image, which we all do in the hospital and Translational Research Institute. In addition to staining, we can use NGS sequencing the proteins or the RNA so we can understand better and correlate the results to our staining images. So this is what 10x provided two main pillar platforms, or the three that we have. Now, so today I'm not going to talk about all the examples. So this is an older slide, but now with 10x, we have more than 5,000 publications that you can access from the website. And actually, many of them are coming from oncology or immuno-oncology. So there are many examples being done in cancer, as well as in research-based or translational. Translational means they use our research-based products, and then they make some projections of what is happening to treat cancer, for example. So all those examples are available. So we invite you to visit the 10x website or just contact us or Pandu Bioscience, and then we can give you some examples of what people have done. So I'm gonna start with the single cell experiments, the single cell products. So one of the key use on the single cell in cancer research is to look at how heterogeneous or look at specific cell populations that we're interested in cancer treatment, for examples. So bulk analysis, as my colleagues just talked about, if you do bulk, you isolate thousands to millions of single cells from a piece of tissues, you get one data point. You actually cannot distinguish all the cell types, and then you cannot find biomarkers because you don't know where they're coming from. So this is a very old study in 2017, 10x single cell started in 2016. So I like these applications, this example, because it really showcases why you want to do single cell. So they study on EGFR in cancer treatment. So they study a drug actually giving to the treatments. So their goal is just to identify the cell populations that responds to the treatment. So in order to be treatment responsive, this cell has to express EGFR. So this is the single cell results in 2017 that they published. You can see from this data, using single cell assay, they were able to easily identify these EGFR responsive populations out of all the tissues that added. So even though this example is pretty simple by now, but really showcase why people want to do single cell, because it allows you to look at, for example, in this case, will be treatment responsive cell populations to therapies. Now, the 10x assay is built not just to measure the gene expressions. You can also do T cell and B cell receptors. So why would you do this? Because, for example, in cancer therapies, immunotherapies, now we generate a lot of CAR T cells that we put into the tumors, and hopefully those CAR T cells will target the tumor, bind to them. So this is a very popular potential treatment regimes that the world are pursuing. So with 10x applications on those T cell immunocancer therapy, you can actually couple the gene expression data with the T cell, B cell receptors, and therefore you can trace the CAR T cells once injected into a study patients. Now, so this is one of the examples of how single cell and gene expressions is being used to classify cancer cell types. So you can see that each of these dots is actually represent one data point from one cell. If the two dots are close together, most likely they belong to the same cell type. Two dots are far apart, they mean they have different cell types. So you can see that through single cell gene expressions, you can easily identify all the major cell types, including tumor cells, tissue resident cell, immune cell types. You can also look at more granular cell types, helper T cells, a variety of different types, different cell types, as well as a variety of B cells. In these examples, they specifically look at plasma B cells that you can distinguish from these guys. Plasma B cells is important in cancer research because these are the differentiated B cells that they secrete antibodies to target specific cells. So if you're interested in looking at cancer and heterogeneity, single cells is definitely the way to go. Now, so as I just mentioned, the 10x assay is multi-omics in general. You can study many dimensions from each of the single cell. This is another example that really showcase how gene expressions is used to study gastric cancer. So in these examples, they take some samples and they do a single cell gene expressions analysis. Now, so for the goal of this one is a lot of people are using 10x assay to build cancer atlas. Atlas means, okay, so I have this gastric cancer, and you want to use single cell assay to look at what cell types is present, how many of them, and what are the proportions. So in this one, specifically in this paper, they want to identify biomarkers for the cancer-specific cells. And in this example, you can see that they were able to identify a whole bunch of major cell types, including those involving cancer. These are the purple ones. These are the cancer cell types. And so by doing gene expressions, comparing the expression profile of this group versus the other groups, they were able to identify biomarkers. So for cancer researchers, single cell is a very powerful tools for you to identify biomarkers that are relevant to, in this case, on specific cancer cell types. Now, so as I just mentioned, with a 10x assay, in addition to gene expressions, you can also assemble T cell and B cell receptors from all the T cell and B cells. And this is very important for cancer immunotherapies. So the data look like this. So what you do is you can sequence the gene expressions each of the cell to classify cell types. If it is a T cell, these are the B cells and the T cells. Simultaneously, you can also assemble and sequence a full-length T cell and B cell receptors. So how is this being used? This is nicely demonstrated in these papers published a long time ago, 2018. This is actually done by researchers at University of Washington in the US. So they studied skin cancer. So what they did was they actually engineered these CAR T cells that recognize the tumor oncoprotein. So they injected those CAR T cells into the patients and then they follow them over time. Now, what they found was some of the treatments are successful in some of the patients, but a small number of patients, they have relapse. That means the tumor come back. So then they use the 10x assay to sequence the gene expression profiles and also assemble T cell receptors. Remember, these are CAR T cells, so you need to know the T cell receptors and trace them on the patients that become resistant. And they start to ask, why are some people become resistant to the treatment? So the data on your right is actually look at the tumor-infiltrated immune cells. So these are T cells that go inside the tumor. They sequence the gene expressions so they can look at different cell types, and they also sequence the T cell receptors. What they found are these are the red ones. So the T cell receptor, the sequence, okay, these are the CAR T cells that are injected into the patients. So they are present in the patients that relapse. So then loss of the CAR T cell cannot explain why they relapse because the T cells clearly go inside the tumor. So to the gene expression data, they look at all the genes that are expressed, then they tune in these HLA expression genes. They found that actually the tumor cells somehow repress the HLA gene expressions. And therefore, this means that even the CAR T cells that we engineer target the oncoprotein can be going inside the tumor. It cannot express the HLA gene. So it cannot do the function it's supposed to do. So the good news is that loss of the HLA repression is known to be reversible. Now, this paper is published in Nature Communications, a high-profile journal, because this is one of the rare occasions that we identify tumor can suppress HLA gene expressions as a way to tumor immunotherapy. And the way to treat this patient potentially will be a reversible regime so that the HLA is not repressed. Now, so I like to show these examples because even though 10x product is research-based only, in translational applications, for example, in CAR T and cancer treatments, there are quite a lot of examples that where 10x products can make really translational application. In this case, will be a great evidence find what happened to tumor that get relapsed with CAR T treatments. Now, the other things I want to talk about, as my colleagues just implied, in addition to gene expressions, you may also want to profile the epigenomes. These are the DNA regions that are open, these including enhancer, repressor, promoter. So these things regulate gene expressions. So this is called ataxia, which you tag open chromatin regions and you sequence them. So this is an earlier paper conducted by researchers at Stanford University in the US. So they studied basal cell carcinoma as well. So what they did was they looked at the open chromatin regions using a 10x assay. So this is really PD-1 treatment regimes. So they were able to use epigenomics to classify all the major cell types. So epigenome is a great way to look at cell trajectory. That means initial, these are the cell type, how they differentiated into the responsive patients. So they found that two groups of exhausted T cells and helper T cells is relevant to responsive PD-1 treatment. And then using the ataxia, they were able to look at developmental trajectory of all the major cell types that are responsive to treatment. So the key for this is not to dive in too much on the paper, but just to let you know that, the epigenomes is also important for cancer responses. So in addition to gene expressions, you may want to look at the epigenome too. Epigenome is kind of the switch that control all these lights. Light is expressed genes that we can measure with gene expressions. But the switch or the controller that we turn on when we come into this room is actually the regulator and the epigenomes. So at 10x, we're actually the first commercial products in the world that you can profile simultaneously the gene expressions and also the epigenome together in each of the cells. Now, the second pillar of 10x technology is the Visium spatial technologies. This is where you profile the gene expressions or the protein expressions on the tissue and link to the histology. So the histology could be an immunostaining image that you have, or in this case, H&E staining. So how is this being used? Why Visium spatial technologies is important? This is a publication in Cell Reports. There are two pieces of tumor, A and B. So on A and B, by eyes or by staining, you cannot distinguish the tumor microenvironment. But with Visium, where you can make the gene expressions on the tissues, in this case, we localize the tumor-infiltrating immune cells. The red dots means the key genes for those immune cells that go inside the tumor highly expressed. You can see that in tumor A, the immune cells actually went inside the tumor. This is what we want to see. That means, okay, good. Immune cells will go inside the tumor. There is a chance that they may bind to the cancer genes and then kill them, hopefully. If you can see this piece of tumor B, the immune cells actually located outside the tumor. They could not go in. So that means whatever we do on this treatment is not going to work because the immune cells cannot go inside the tumor. So you can see by these examples, even though this looks simple, spatial biology is really important tools for you to look at tissue biology, right? Whether my immune cells go inside the tumor or not. This is going to be the first thing that you need to look at in your tissues if you're looking at immune cancer therapy. So this is an example, again, published a long time ago using the first versions, the initial versions of our Visium technologies. This one, they look at prostate cancer. 12 different pieces was sent to a pathologist for eyes annotation. I want to tell you why Visium is a great complementation to what we can annotate by pathologists. In the top, this piece of prostate cancer tissues. So we do gene expressions on the tissues, and then we were able to find the cancer genes. This is the same to what we can see by eyes, annotated by pathologists in this red dot. But if you look at this second piece, by eyes, the pathologist could not see any cancer cells. But by NGS gene expressions, you can see that we actually have some cancer cells present in this one. So this really highlights why Visium spatial technology using NGS is so important. This is a great way to what you might be doing in the hospital, using eyes as a traditional way to annotate cancer tissue biopsy samples, because there's only so much we can see by eyes. But NGS really can enhance or supplement what pathologists can do. So, and again, if you have any pathologists in the audience, if you're interested in what Visium enable you to have a more precise way to look at your tissue cancer samples, one more examples, talk to us. We have tons of examples that are published or conducted in-house that showcase these kind of examples as a way to hopefully, as a routine use for you to do pathological annotations in biopsy samples or cancer tissue samples. Now, today I talk about single cell and spatials. These are not independent technologies. In fact, they can be integrated. I'm going to show you an example published in Cells, how powerful integration of single cell and Visium spatial technology is. So this is another example. They studied skin cancer as well. So what they did was these researchers, they took some of the pieces, tissue samples, and do the single cell assay. The goal is to look at cell types that are relevant to skin cancer development. Some pieces go into Visium. So first, they did single cell study. Again, they were able to identify many major cell types on the skin cancer. They found one cell type that's interesting. These are called the tumor-specific keratinocyte. These kind of express invasive behavior by gene expression. That means these kind of cell type, they express some genes that seems like they're cancerous. They want to invade other regions. So then obviously, the researchers is very interested. Okay, so I know these cell types, but where are they on the tissues? So they use the Visium, looking at tissue biology, looking at the gene-specific marker for these cell types and localize them on the tissues. Through Visium, they were actually able to find that these tumor-specific keratinocytes, they're located close to our blood vessels. So what it means is that combining in single cell and Visium, it means that, okay, so first two single cell identify new cell types that are relevant to skin cancer development, these specific cell types. To Visium, they localize close to the blood vessels. So in theory, maybe these cancer cells can easily go inside to our bloodstream and then circulate and invade other regions, and this is potentially how the skin cancer can spread. Now, so I really love these examples because this really highlight the complementarity or integration of a single cell and also the spatial data to help you to have a more in-depth overview of what is happening in a cancer biology. Now, to summarize quickly, so today I talk about two main technology in 10x. Single cell allows you to classify different cell types, looking at cell types that are relevant to immunotherapies, looking at biomarker of specific populations. The Visium spatials allows you to visualize where all these biomarkers are present on the native tissues, and you can link the NGS data on the Visium to pathologist annotations. Now, many of you, I'm sure, that are doing pathological annotations in biopsy slides, do think about adding on the NGS dimension. As I showed earlier on the prostate cancer samples, NGS can really enhance or improve a lot of pathological annotations on where you can see the cancer cells or what potential stage is the cancer is using the NGS and Visium spatial technologies for annotations. So with that, thank you, and we'll be happy to take questions later, right? Yeah, thank you.
I will invite all the speakers to speak. We will proceed to the question and answer sessions. Please, Dr. Sean, Dr. Yanti. And before I will check first if there is any question from the audience in the chat room, or there is any question from the audience in the room, you may. Please, Dr. Putri, please.
Thank you, Dr. David. Yeah. Hello, San and Leo. Thank you for coming to our hospital. It is an honor for us. Actually, I want to ask one question to Sean and one question to Leo. So, for Sean, what is the unique value of single cell RNA sequencing compared to standard RNA sequencing that we've already run previously? So yeah, maybe before you answer, I just go to Leo. So for Leo, what is your advice for pathologists who will have a first experience for single cell technology? Thank you.
So I think, thank you for your question. So the first thing is that, when you do bulk sequencing, bulk RNA-seq, you're extracting RNA from a piece of tissue. And from my understanding, in an operation theater or when they're performing needle biopsy, they'll try to get the cancer tissues out of the patient, but at the same time, there will be also regular tissue type. The percentage of normal tissues, the percentage of cancer tissue is hard to determine unless you have a trained eye. Even with the trained eye, they're looking at the protein staining, the IHC results to see those are cancer. And at least we see from the RNA data set, we saw that there are times where you will miss out certain morphology because the stain doesn't stain because there's too little protein or cell types around that region. And you're just getting an overview when you're doing a bulk. So everything gets mashed together. You get one output. You still look at all the 28,000 genes in the human, but the expression profiles will change if you subset them out into single cells, because you will be able to identify the signals that comes from the cancer cell. You'll be able to get the signal that comes from normal cells. And normal cells, honestly, those are not going to be the target for you when you're designing drugs against cancer cells, because you're trying to kill cancer cells. You're not trying to make the normal cell grow more or faster, because then they'll become cancer. So if you do bulk, some of those signals itself may be attenuated, because if you may have to determine where the signals are coming from. But with that said, bulk sequence, usually those high elevated signals itself is already above the norm. So you can see them. But what is dangerous is you assume all the cancer cells are the same. A solid tumor itself is made up by a lot of microenvironment, a lot of tumor microenvironment in there. And if you design a drug simply just based on the most elevated signal, it could come only from a subset of cells in it. And having such a harsh treatment may result in a poor survival rate when you administer the treatment. That's why I think when you perform cancer therapy, they try to regulate the dose and increase it accordingly. They don't start off right at the start as the highest dose as possible, because the patient probably will not survive the treatment rather than survive the cancer. So with single cell, you may be able to identify what kind of treatment, what kind of cancer variety cell types is in there. Some of them may be more, maybe like in the breast cancer, it could be a single positive, it could be a double positive, it could be a knockout, it could be a triple negative. Normally, we are only based off the histone or the biopsy to determine what is whether how many positive, how many negative is in there. But it might turn out to be, as we understand further, it might turn out to be, it might be different tumor core, maybe double positive, some a few maybe triple positive. And that is something that I think that we have to look deeper into to see whether it can help to affect. But with a bulk, you're not going to see this.
Great, thanks. So the questions will be my experience with single cell. Yes. So so on the 10x single cell products actually came out in 2016. The gene expression first. So now 7 years later. So what do you know? So what we can for sure say now, single cell technology starts to become a norm in all worldwide. So you can see with the 10x technologies alone, 5,000 publications, most of them can be single cell actually. A lot of journals and publications or even translation research now relies on single cells to interpret that data. So bulk sequencing is definitely going. The microarrays is really the first phase. Then you go to NGS-based technologies, but NGS-based is one point. So a lot of people now recognize that getting on each of the cells worth of data compared to bulk RNAs is the norm. That means they require you to do that. Now, some of the examples that we showed throughout today, you can see, the whole, if you do one RNA sequence point, you got one data point. And those things are usually cancer responsive, or you look at specific cell types of cancers, are only a small populations among the others. And people recognize single cell as a super powerful tool to look at heterogeneity. So single cell has been adopted quite. But your questions will be, how do I start? The key things is actually on cell prep and dissociation, which Sean will help. Honestly, with the technologies that now we have 7 years later, there are so many improvements, guidance on the web as well as 10x, that in many major cancer cell types now it's easy for you to just go to the website, search for publications, replicate their cell prep protocol, talk to a 10x. We have a lot of experience now over these 7 years working on it. So to me, the key things will be just to start. So, sometimes it's very hard to convey to the funding agency and say, honestly, why, we want to do this and this is important. We have many examples that people can really see things that they cannot see before, which is very, very important. So to me, the key thing will be it's very hard to start. And I think the same in Southeast Asia, a lot of countries, but once you have it initiated, we will help you. On overcoming the challenges, mostly in sample prep, is very possible with 10x because we do have support on the bioscience also provide support. And I strongly encourage you to really adopt the technologies now because we are very confident that over these many years, single cell is a norm. That means it is going to be needed for you to look at biological complexity. It's potentially it's just like a PCR instrument now. Single cell is people use it for all kinds of uses. So so the key thing is do not worry. We're going to help you. You have the initiation to start. So we're going to help you to prep the samples, run it, even assist you with sample, with look at your data, how to do it. One thing I did not mention is that with 10x, we actually provide end-to-end solutions. There's instrument reagents. All the analysis tools are on the website. They're very easy to use. So all these are provided by 10x, and so that you're not afraid to start. We can help you with all these tools. So there's no extra cost for you to analyze the data either.
So after the single cell technology, so do you still use NGS or do you still use NGS?
Yes, for single cell assays and the disease spatial as well, you do have to use NGS. So you make it into a Illumina sequencing library. So you sequence all the expressed genes. And so, yes. So what you do is that you put the single cell, put it into the machines, and then you do the workflow bench. And then at the end, you make it into a library. You send Illumina, sequence them, and then you use the software tool to analyze the data.
So you still need bioinformatic analysis from the 10x or?
Yes, but I'll give Sean some time to talk about this.
The bioinformatic analysis, the software from 10x is free of charge. So you can just download the analysis pipeline, depending on which kind of data set you're working with. There'll be cell ranger, space ranger, Xenium ranger in the future. However, to handle those large data sets, my recommendation is to get a specialist bioinformatician who is familiar with handling single cell or large data set, because what we can do is just a preliminary data analysis. It may not be customized to fit everyone's questions. So that will still require the third-party software or the trained bioinformatician to answer the questions that you have. Like how did the cell develop from normal to cancer? There's a mixed pool of cells. How did T cell plurality expand from which T cells? That requires, again, another type of analysis tool to project. But the initial data is all generated from the 10x pipeline. So you don't really need your own in-house pipeline to do the first step in data analysis.
So all in.
It's all in there. Yeah, in fact, we don't charge them. The charge is actually goes to your kit.
Okay, thank you.
So one thing I want to add is that I travel quite a lot, especially in Southeast Asia as well. Data analysis could be scary, but honestly, do not worry, because a lot of researchers outside Indonesia, they want to collaborate with you. I have actually linked many successful collaborations. If it's okay for you to send the data. There are a lot of people, the closest one in Singapore, Australia, we have linked a lot of people together. They help you to analyze the data. So during the process, you may not have to set up a report, and then they also help you with processing it and helping you to analyze the data. The only thing most of these people ask from those other things is to put their names on your papers, honestly. And so if you have decided to say that, okay, but I have many challenges, I need some time, but I need help, do not worry, talk to us. We have a lot of researchers outside that from universities, so they're okay with collaborations with academic universities, and then they will help you. We have many successful collaborations like this to help you and to set up. So it is a great collaboration. If you need anything, just talk to us. We can introduce those people to all of you. Yes.
Is there any question?
Do you have anything to add?
Dr. Yan?
No.
Okay. While waiting, I will ask some questions first. We're dealing with large number of samples that are stored in our archiving. So can you give us any experience how if we use FFPE samples, how old the samples will be good to be to be examined with the technology, especially the efficient spatial?
So I'm excited when I hear that you have archive samples. So I assume those are archive FFPE blocks. There isn't a real hard and fast rules or cut off to the age of the block. It's more on the DV200 value. So if you have a block that's been stored for even 15 years, I would suggest section it out, take a look at the morphology first, check whether are they all dried up or are they all overfixed. because some of them can darken and become flaky. So those kind of blocks itself, even for your IHC, is not going to work well. So if the morphology you perform H&E, it looks intact, it looks fresh, it looks clean, and then you can check the DV200 values of the RNA quality. If the percentage is higher than 30%, I think it's worth a shot to go forward. In-house, why are we recommending those values? It's because those are the ones that we have tested in-house with the kind of minimum 30% and above that has a higher success rate of getting usable data. I'm not going to say that it's going to be definitely successful, but at least there's a higher chance of getting usable data. With that said, we do have customers in Australia that have used block that is slightly over 10 years, and they were able to generate a really good data set. And this is even with the direct placement assays. So end of the day, I would say check two things, just a simple QC, check the morphology, do a H&E stain, see whether they look, you have the structures, it's still there. Because if the structure is all gone, I'm not going to waste my time to do a DV200 because that is a chip that will be wasted. Right? I always look for the easiest, cheapest way to QC first step, followed by my second QC, and that'll be looking at the DV200 subsequently. If you happen to have OCT archive, frozen tissues in liquid nitrogen, then you can do the same thing. It's just that instead of looking at DV200, you will be looking at RIN value. So that will be 4 or higher, so above 3. So just to add, if it's fresh frozen, all right, if you realize that the RIN is low, try to fix it and then test it with the fixed frozen and check. Of course, that you can check with the morphology again and then check with the DV200. If it's above 50%, it's worth the try.
So I will check if there is any question in the chat box. Yep. Maybe from the audience again, if still there is a discussion or comments about the products. We still got 5 minutes, yeah, I guess, before we can close the session. Okay, so the Q&A session is concluded. So thank you very much for our speakers for sharing the knowledge, and hopefully the technology will can be incorporated soon in Indonesia. We hope we as Darmais can be the first to use the technology. And we give applause to all the speakers today. Is there anything to add? Okay. Okay, we will have the photo session. I invite all the online participants to turn on the camera. Okay. We will wait for someone to join.
I can't see anyone wave back, but no.
The participants are actually joining from across Indonesia. I see some from Sumatra, from Riau, from Bali, and is there any? Okay, can we proceed to the photos, taking the picture? Is that? Yeah. Okay. Ya. Oke, para peserta sekalian dalam hitungan yang ketiga. 1, 2, 3. Cheers. 1, 2, 3. Terima kasih. And then we'll take okay. I invite all the speakers and the 10X teams to join us in okay, all the participants may join to the photo session. Take a picture and on.
Ini di dulu nih. Ya, itu nanti kesandung.
Mau ada mana? Anak mana?
Kalau di sini backgroundnya itu.
Kalau background ini
Backlight.
Sini mungkin. Tapi momennya ini ya. Tapi momennya
Kalau backgroundnya.
ya coba aja sini tuh ya. Yang belakang sudah kelihatan. Iya. Harus dipotong. Nanti coba ya. Ya, itu betul ya. Ada langsung. Tinggal pasal dulu. Orang nanti. Kontak. Boleh. Oke, coba.
Para peserta sekalian, acara kita telah selesai. Terima kasih atas partisipasinya. Semoga acara ini boleh bermanfaat bagi kita semua.
Beneran nggak selesai?